Biology starts in a distant past and it points towards the future. With this notion, I greet a very good morning to one and all present here, and I wholeheartedly welcome everybody to the International Virtual Conference on Biology in 21st Century, organized by the Department of Zoology of Konganadu Arts and Science College, Coimbatore, Tamil Nadu.
Prayer is a positive vibe. The good start always begins with the blessings of the Almighty. So I pray the Lord to shower His blessings throughout the entire session. The time has come to ravishingly welcome you all to this program. Now I call upon our respected Assistant Professor of the Department of Zoology, Dr. B. Mayalagan Sir, to deliver the welcome address and also to introduce and welcome our today's Honorable Resource Person, Dr. Naik's Research Scientist, ICGEB, Emory Vaccine Center, International Center for Genetic Engineering and Biotechnology, New Delhi. Please, sir.
Thank you, Nishandini. Am I audible?
Yes, sir, you can
Yes, sir, you're audible, sir.
Yes. It's very good morning to one and all gathered here for this today's session of International Virtual Conference on Biology in 21st Century, organized by Department of Zoology, Konganadu Arts and Science College, Coimbatore, Tamil Nadu. On behalf of Department of Zoology, Konganadu Arts and Science College, I welcome the most respected President, Dr. M. Aruchami sir, most respected Secretary and Director of our college, Dr. C.A. Vasuki Madam, respected Principal, Dr. M. Lakshmanaswamy sir, and most dynamic former Vice Chancellor of Tiruvallur University, former Registrar of Bharatiyar University, K. Murugan, currently Emeritus Professor in our department, Dean R&D, Dr. S. Palswamy sir, Dean Academic, Dr. S.R. Madan Shankar sir, and Head in Charge, Dr. S. Veenakumari Madam, fellow colleagues, participants, and my dear students. It is my honor to welcome today's resource person and introduce him to this session, Dr. K. Kaustu Nayak, Research Scientist and Immunologist, Emory Vaccine Center, International Center for Genetic Engineering and Biotechnology, New Delhi. Dr. Nayak completed his bachelor degree in zoology and a master degree in biotechnology at Loyola College, Chennai, and he obtained his doctoral degree from Tuberculosis Research Center, ICMR, and the degree was conferred by University of Madras. Previously, he worked as a research scientist 1 in the Tuberculosis Research Center and ICMR Centenary Postdoctoral Fellow at Institute of Research in Tuberculosis. He has published over 20 research articles in highly reputed journals, and he has a co-principal investigator of project entitled Functionality of B cells in dengue and chikungunya co-infections, funded by Department of Science and Technology, India, about the amount of rupees 56 lakhs. Dr. Nayak is also worked under the guidance of Dr. Soumya Swaminathan, Chief Scientist, World Health Organization. And it is our pleasure to have you, Dr. Nayak, to this part of our virtual conference. Now, I hand over the session to Dr. Nayak. Thank you very much for the opportunity given to me. Now, it's a time to Dr. Nayak. Thank you very much.
Thank you. Thank you for the lengthy welcome. And thank you, Dr. Manivandan, for inviting me. And it is my pleasure to talk to the audience here with special invitation from Manivandan, who happens to be my PhD batchmate. So, like, how do I just go and start the presentation? Like, do I need to click something here?
So you can start your presentation, sir.
Yeah. So let me just tell you, even before starting, especially for the students, you can probably stop me in between, ask me questions. I don't want it to be a one-way presentation. And so I have also told Manivandan regarding this, that after this session, if you have questions which need not be particularly to this presentation, if you have any questions about career in science, career in life science, what to do after you do your master's or bachelor's, how you look forward your career in this field, you can definitely ask those questions as well. And And yes, you can stop me in between. So this is, take you to the technique I'm going to speak to. I wanted to tell you little about basic immunology. I doubt if zoology students have any papers in immunology. So I thought I'll give you a little bit of brief introduction in immunology. And before doing that, I want to start with telling about the institute I come from. The institute I come from, as you know, was introduced that was as International Center for Genetic Engineering Biotechnology, situated in New Delhi. It is a United Nations organization under UNIDO, which started in 1994. It is mainly in 3 different countries, that is India, New Delhi, South Africa, we have it in Cape Town, and then we have one center in Trieste, Italy, which is the headquarters. And there are more than 60 countries who are the member states. So how it works is that each member state's GDP, according to the GDP, they pay for their country's ICGB, and then that's how each country contributes and runs the institute. So all these member countries are eligible to send their researchers and scientists to work in these 3 centers. And I'm giving you an introduction about the institute because all of you can think about ICGB as a place where you can start working for your doctoral program or any other research program, not only in Delhi, but in Cape Town as well as in Trieste. And there are many fellowships available for Indian scientists to work in the other 2 centers. So these are the 3 centers where ICGB is located. And ICGB New Delhi component has more than 25 research groups and more than 100 PhD students. So you can look forward to work with us. You have to just come to us with a fellowship. ICGB has an entrance exam after your master's. So get your ICMR, DBT, DST fellowship, and then you're cleared the ICGB entrance exam, and you can you can enroll yourself for the PhD program. My lab is called the ICGB Emory Vaccine Center. So Emory Vaccine Center, as you know, is one of the foremost pioneers in vaccine research, situated in the city of Atlanta, US. And ICGB and Emory Vaccine Center have come together to start this very unique collaborative venture almost 10 years from now, where our foremost idea was to look into the basic, any kind of infection, any kind of health issues of local problem, that is of India. We wanted to bring ICGB, using ICGB's expertise, using Emory Vaccine Center expertise, put them together and bring them into Indian perspective. So that's how Emory Vaccine Center, ICGB, was born. This is my extended family, my lab. The gentleman in right here, he's the director, Emory Vaccine Center, Dr. Rafi Ahmed. And she is Dr. Anmol Chandel, who heads the lab here.
Sir?
Yeah.
So I don't think so the presentation is visible to us.
Oh, so is there a time delay?
You have to, no sir, you have to present the screen, I think so. Did you do it? In Google Meet there will be an option called
mode.
Okay, sir.
You're able to see now?
No sir, it's not visible for me, but let's ask others too. Is it visible?
Yes, sir. It's
No,
not
no,
visible.
sir, it is not.
Yeah, you should have told me. That's what I asked you, right? That should I click something? No one stopped me. Anyway, what I'm supposed to do?
Sir, there will be an option called present the screen in Google Meet.
Present now, sir. Yeah.
Okay, you're right.
Yes, sir, it's visible now.
Yes, sir. It's now
Oh,
visible.
thank God someone stopped me. Yeah, so anyway, with the introduction, I'll just run through the few slides that you missed. This is the 63 different members I was talking about, the 3 places where we have ICGB, then this is my lab. Next, I would tell a little bit about immunology, basic immunology. I don't know, a few of you might have some what you call immunology papers, but majorly I know zoology and botany students do not have any immunology papers. So basically, we know there are 2 types of immunity: innate and adaptive. Innate is the first line, which we are born with. But then adaptive or acquired immunity, this is where it is acquired later. So innate immunity is something like, as I told you, is the first line of defense. Many of you might remember a very old computer game, the Prince of Persia, where he needs to fight different multiple walls, and to save the princess. So think about like the Great Wall of China, which was built to keep enemies off. The same way, innate immunity is the first line of defense, your skin, but they're very non-specific, very, very non-specific. They are molecules, cells, but as I told you, very non-specific. They don't have any memory. But yes, information can be passed on, just like a home guard or your security in the society who informs that, yes, there has been a rupture, we need to take further action. Next is the adaptive immunity, which is the second line of which is more specific, where some amount of memory is involved. It also depends on the genetic events and cellular growth, and it slowly responds and grows with age. And yes, as I told you, it has memory. Adaptive immunity again is divided into natural and artificial. For example, natural can be acquired by any kind of infection, for example, dengue, which I'll be talking in detail in later part of my talk, and artificial by vaccinating. Most of us are vaccinated for COVID now, I presume. But yes, some of us has some amount of natural immunity to COVID. We know, and yes, if anybody has any specific questions for antibody response in COVID, you are welcome. Adaptive immunity has again 2 parts: the cellular immune response, in which we have the T cells, and then cell-mediated immune response. And these are very specific. Here, as I told you, the T cells, where, for example, you can relate to the army, which goes, detects and destroys. Cytotoxic T cells are responsible for detection and destroying. The next, the humoral immune response, where the B cells are involved. They are the antibody mediated. The antibodies need to travel along, just like for example, the B-53 bomber. You know a specific hideout of the militants. The B-53 bomber goes, destroys, and comes back. In the same way, antibodies are sent to the area of infection that destroy the infection. So I'm giving you this analogy because it's very easy to remember. Now, with this basic immunology background, let me take you to what we do in our lab. Focus of the ICGB Emory Vaccine Center is to look into human immunity to infectious diseases, and especially dengue and chikungunya. Although presently I personally also study tuberculosis with my background in HIV and tuberculosis from ICMR Chennai, and presently we are also working on COVID. So here at ICGB Emory Vaccine Center, we study phenotype of plasmablasts and plasma cells. We study memory B cells. We have successfully been able to produce monoclonal antibodies for dengue, chikungunya, and COVID. We study phenotype of CD8 and CD4 T cells. And then, of course, we study their functionality. As you know, in the response to any specific antigen, the ultimate goal is to develop a immunological memory. Let it be by vaccination or let it be by any natural infection, which will take place either by humoral immune response or by the cell-mediated immune response. One mediated by the B cells, the other mediated by the T cells. So let us look into the B cells first. So when there is a, say, someone gets vaccinated, the immune system gets sensitized to produce the antibodies for that antigen. And slowly, as in later, if part of the vaccinated individual, the actual pathogen is encountered, there is sudden upsurge of the antibody response by the recall response, and the pathogen is taken care of. We study this. How do we study? There are certain plasma cells which we all know as plasmablasts. As the name says, plasma and blasting, so these are blasting cells, but they are plasma cells, they are antibody producing cells. But the catch is that these kind of cells are found in circulation of the circulation only during a very small window period, that is around 4 to 7 days of any viral infection. So if you don't catch these cells during those 4 days, we miss it. So and plasmablasts are very unique B cells which actually give you a snapshot of what's happening in the patient's body during that time. Whereas memory cells tells you what happened earlier. So we study both the memory cells and the plasmablasts. Plasmablast tells us what's happening now. Memory B cell tells you what happened earlier. And these plasmablasts are found in the circulation at very, very high frequencies. So these plasmablasts are B cells. We have seen patients in which almost 100% of all B cells producing antibodies are dengue specific. So by studying this plasmablast, we can actually tell the disease status of the patient. And most of the cases, the plasmablast frequency parallels or matches with the severity of the disease. So if the patient is very severe, you will find very high plasmablasts. And these plasmablasts are cells which we isolate by using flow cytometry to make monoclonal antibodies. This is just what you call preliminary data we had to show that during acute dengue, what is the frequency of plasmablasts compared to healthy individuals versus convalescent, that is after those 10 days of fever. They immediately come down. The next technique which we use is Alispot, as the Alispot stands for enzyme-linked immunospot. Many of you might have read ELISA. The Alispot is a little different for ELISA, as in ELISA you have a very soluble substrate. The end product is a colored reaction. But in Alispot, it's an insoluble substrate which precipitates and makes a spot on the surface of the membrane. In ELISA, we use a plate, whereas in Alispot, we use a PVDF membrane, a charged membrane. In ELISA, you can measure. It's a quantitative measure, semi-quantitative or a quantitative method. But in Alispot, it is it's semi-quantitative. You can measure and just say if a particular cell is producing a particular kind of cytokine or an antibody and how many cells they are. And it's very specific. Even if one cell is producing a particular antibody, an Alispot can detect it, whereas ELISA you cannot detect. And these Alispot techniques are they're very much used and very commonly used in vaccine studies, because say for example, a cohort has been vaccinated for a particular disease. You want to see if they have already started producing antibodies. Alispot is the first technique a researcher would do to look into the antigen-specific antibody which is being produced. So what we do, we coat the membrane with the antigen of interest. In this case, if they are vaccinated individuals, we coat it by the same antigen. It could be a viral protein, it could be a bacterial protein, or a parasitic protein. The blood cells are separated and added onto the cells. They're incubated overnight. So if there is any amount of antibody which is being produced by those vaccinated individuals, they would bind. Then we do the IgG bioattenuated antibody and then the substrate. And these are very specific, experiment technique. You cannot go wrong in this. And if there is a antigen specific stimulation, you will definitely see the production of the antibodies. So the specificity can go up to 1 cell in 1 million. So that means even if there is one cell which is producing a antigen specific stimulated cytokine or antibody, you will be able to detect it. Next, we are interested in looking at the antibody functionality. There, our lab has been pioneers in producing human monoclonals. This technique is little different than what we all studied during our BSc and MSc by hybridoma technology. This is this is not hybridoma technology. In this technique, what we do is we sort plasmablasts. As I told you, the plasmablasts are the B cells which are producing the antibodies. And these plasmablasts are antigen specific. In this case, they're dengue specific. So what we do, we isolate dengue specific plasmablasts from the patients using flow cytometry, or FACS, what we all know as. Then many of you might have studied the antibody genes, the heavy and light chain. So they are cloned, and then we transfect these antibody into a 293 T cell lines. So this technique has been first established in our own sister lab in Emory Vaccine Center in the Atlanta, and we brought the technique to India, and now we have established it here at ICGB Emory Vaccine Center. So we have, as I told you, we have produced antibodies for dengue, chikungunya, and COVID. And many of these antibodies are neutralizing in nature, and they could be used for therapeutic purposes, for treatment. So this is the basic schematic representation of how we produce these antibodies. So we take B cells. CD19 is a marker for B cells. So we take the B cells, CD27 and CD38. They are receptors which tells you that they are activated cells. So we take activated B cells, sort them into 96 well. So 1 cell per well. After sorting 1 cell per well, we do, as I told you, we clone the heavy and light chain and produce it by using basic cloning technology. Next, looking into the cell-mediated immune response. Looking at CD4, CD8 responses. This is the basic immunological picture which all of us might have seen in any immunology textbook, how an antigen is presented by that CD and CD8 cells, along with the HLA class 1 and class 2. And this is the same thing we use to study the T cells, which are CD4 and CD8 T cell responses. And one technique what we do is epitope mapping. So many of us have studied what is epitope. Can anyone can tell us what an epitope is? Anyone? I mean, I just want to know the level of, because I don't want to go overboard by talking lot of immunology terms. Can anyone tell, anyone who has studied what is an epitope?
So it's a part of an antigen molecule where antibody goes and attach itself.
Right. So by epitope mapping, we just want to see what is the part of the epitope which is getting recognized. For example, in this, what we do, epitope, these are peptides. Peptides are, every antigen is, every epitope, it's the building material is amino acids, right? So what we do, we break down a particular peptide sequence of, for example, any peptide sequence of a antigen into overlapping amino acids. For example, in tuberculosis, say there could be 4,000 different amino acid sequences, but you cannot study all of them at one go. I mean, studying T cell or B cell responses, what do we have to do? We have to use these peptides to stimulate the blood cells and then look for their which of the antigens or which of the peptide sequences actually stimulate. So by stimulating the blood cells, we'll understand if the T cells or T cells are getting, I mean, T cells are recognizing them and are producing any inflammatory cytokines or not. But you cannot study that many amino peptide sequences at a time. So what we do is we do a simple technique known as epitope mapping. In epitope mapping, what we use is we use overlapping peptide sequences. We mix them in such a manner that all the peptides are we mix them in such a manner that each of the peptide remains intact without masking the next one. For example, I'm giving you a matrix here. We have mixed these 4 peptide sequences in such a way that peptide sequence A, B, C, and D, I mean, pool A, B, C, and D, 1, 2, 3, and 4 would not mask each other. But we have reduced the number of peptide sequences which will be used for the experiment, but still kept the peptide sequences numbers intact. Here, we do both for dengue and chikungunya. I'll show you a little bit of chikungunya data here. By flow cytometry, we found that especially, for example, E3 protein, that's E3 protein of chikungunya gave us a lot of stimulation. But what I'm trying to say is that this is a simple technique which any immunology lab can use to study the immunology, what you call, which particular peptide is immunogen, and then break down those peptide sequences to further use molecular docking and anything for using any vaccine candidates. Next technique, what we do is simple ELISA. I would not go into details of it. I think many of you would have actually studied ELISA. We have done Alispot. Yeah. The other technique, what we do is looking at the memory B cell. The frequency of memory B cells that is circulating in our blood is less than 0.02%. So it's it's just not possible to detect them, study them as such. So what we do, we take out the blood, we isolate the lymphocytes, we culture the lymphocytes with Coquid mitogen, Staph aureus, and CpG. These are the 3 things which will actually stimulate only the B cells. So it's like 0.02%, they would stimulate, and for 6 days they stimulated, and the frequency of B cells become almost 100 times. And then we study the antigen specificity. So from 0.02%, for in this, here I'm just giving you an example of flu or dengue, but you can use for any antigen of interest. You can do this particular technique to study the memory itself. Next, of course, we do the flow cytometry that is almost on a daily basis. Flow cytometry is a process where uh, how many have you studied? Anyone has studied flow cytometry? Even, I mean, theoretically.
Yes, sir. So I think so. I have a little amount of practical knowledge in flow cytometry.
Oh, great. So yeah, so flow cytometry, as we all have, we all call it as facts, but yeah, its basic technique is flow cytometry is a process in which the chemical character or physical characteristics of cells are studied based on by using antibodies. So as the name says, flow, fluid, cyto, the cell, and metry. Metry means studying cells in a fluid. And of course, the flow cytometry has reached very high sophisticated manner now. Probably when I started doing flow cytometry in 2001, we had only 4 color flow cytometry. Now we have reached at least 100 colors. So it's very sophisticated. It has reached very high sophistication. So by in a single tube, you can learn everything about the cells. Now, with these, we study the problem of dengue and chikungunya. This is a very common picture of what you call site of any hospital in New Delhi during the dengue period, which has already started. And when it comes, it's like you see patients all over the hospital. This is a picture probably of All India Institute of Medical Sciences or any other government hospital where we get overwhelmed by the patients. We get overwhelmed because we get blood samples from them to study the basic immunology. Dengue, many of you may not know that India is a dengue capital of the world. That's huge. We have around 390 million cases. Huge. But due to many other reasons, the burden of dengue is somehow underestimated. We miss them. So it's very difficult because what I personally see at the hospital is overwhelming. And we, as immunologists, we try to support the clinicians at multiple hospitals in Delhi to take care of the patients. And if you see this is India. This is a particular kind of graph which is made based on the burden of infection of that particular disease. And with this, you see the size of India is huge here. That's because the number of cases, that is number of dengue cases per year. We get at least 30 to 40 million per year. Next, going to chikungunya. Chikungunya, we didn't have much. Chikungunya was first seen in 1964 in Kolkata. And then slowly it grew. I remember in 2007 there was an upsurge, 2010 there was an upsurge. So what I'm trying to say is slowly chikungunya used to come and go back. I mean, it used to vanish. Now, every 10 years, every 5 to 8 years, we previously felt that chikungunya, that upsurge of the sudden upsurge of chikungunya occurs due to the imbalance between the naive and the exposed population. Many of us might have studied herd immunity, but now that's not the case. The episode which occurred in 2006 has never gone down. So some things have changed in chikungunya. Many studies have told us that due to mutation in the chikungunya genome, this is happening, but we still do not know. So a lot of work still needs to be done in the immunology of dengue and chikungunya, because that's my personal interest. I study chikungunya immunology at this lab, and so I would probably now finish this, and of course I would like to tell little bit about the Indian Immunologist Society. I don't know many of you know about it. This is the only immunology society in India, and I'm the joint secretary. So I invite you all to be members of the society, and that in that way you will be getting connected with so many other immunologists in India. We have around 2,000 members all over India and abroad, hardcore immunologists, and it will be great to have people who are of basic scientists like you, who have studied zoology, botany, or life sciences, to be a part of the society. Thank you.
My participants are requested to stay with us for a few more minutes. Due to technical issues, our speaker had left, so you will join with us shortly. Please stay with us.
Yeah, I'm audible, right?
Yes, you are audible now. Yeah.
Yeah. No, yeah, so I told you, so students can ask any question pertaining to either the talk or any other questions which they feel about science, about career, about anything. So I'll be happy. So it's, I mean, yeah, I don't think so. You should feel shy if, I mean, what I'm telling is if you find an opportunity to ask someone some question, you should ask. As simple as that. Don't feel shy about it. Any kind of questions. Yeah, so if you're not very think English or Hindi, yeah, you can talk to me in Tamil. I understand Tamil.
Sir, I have posted a question in the chat box.
Let me. So what is that? Let me open the question. Yeah, do you prefer preprints or peer-reviewed journals for your publication? Oh yes, we would. We do. We have recently we have put both our publications from last 1 year in preprints, and then it goes to peer-reviewed journal. Yes, preprints are a way to go now because preprints are. I mean, you can actually cite preprints even before your paper gets reviewed. You can expect few people to cite your paper. So yes, personally I prefer preprints should go before. So did I hear you, Mr. Guru?
Right, sir. I got it. Thank you, sir.
Yeah. No, because you understand, because there have been instances where people put the papers into peer-reviewed journals, and due to whatever reason, the peer-reviewed journal don't accept it, and then you find a similar paper overnight. So this also prevents that kind of what you call malpractice, if you call it.
Sir, is that like இப்போ பார்த்தீங்கன்னா வந்து ஒரு 15 years 20 yearsக்கு முன்னாடி we didn't have these preprints, and we can also see that problem very evidently in the recent book given by Jennifer Doudna, The Code Breaker, where there was a problem in applying for the patent. So do you think so? Do you think that preprints are actually a way to
see patent? See, I very maybe last few months back I was attending an interview in which they asked me the same question. They said, Dr. Naik, you scientists log are not patenting, but of course, now if see, for example, funding agencies, patenting is a very long process. It's not as easy as we think. But if we prefer to patent, then of course we cannot publish it. Then it has its own. It's a double-sided sword, because நீங்க பேட்டன்ட் அப்ளிகேஷன்ஸ் போட்டீங்க, பப்ளிகேஷன் பண்ண முடியாது. பப்ளிகேஷன் பண்ண முடியலன்னா PhD ஸ்டூடெண்ட்க்கு PhD வராது. Many universities are now asking for your papers, first author papers, 2 or 3. So it's a vicious cycle. You have to decide what we want. If you think your science is going to bring out a product, but then you must have enough booty, enough money to go for the patenting purpose. Patenting takes long time also, and many of us have no time to wait, because we are in such a race. Either a scientist needs publications to grow, or for the funding agency needs. So that's my personal view. Because yes, to answer your questions, if you patent and you cannot publish, if you publish you cannot patent. So it's your choice what we want.
Thank you, sir. And I have another question too.
Yeah, go ahead.
Sir, is there any internship opportunities to work with you? Because I'm very interested in working with you, because my area of interest lies in immunology.
Yeah, to answer your question, no, because ICGV has this COVID everywhere. They don't allow visitors. What if they don't allow vendors also? Like Tarson's company fellows cannot come in now. Okay. And ICGB, yes, you can apply whenever, later, that time, whenever, what is the rules that time, but at present, no one, their admin is not allowing anyone.
Thank you, sir.
So let me see, there are some more questions. Combination of Covaxin and Covishield. Yeah. So, yeah, personally, if you ask me as a virologist, I would prefer Covaxin. I was waiting to get Covaxin, but I got vaccinated only 2 weeks back with Covishield, because I couldn't get hands on Covaxin. I would prefer Covaxin, but yes, looks like Covaxin probably doesn't have the primary dose is not eliciting enough immune response, but it will be slow. It's a whole virus, right? So that's why probably, but we'll have to wait, because it's too early to even come to any conclusion. Any studies which have been made now have been done only, say, for last 8 months. It's very early because all these vaccines have been used on emergency purpose only. So we cannot actually come to any conclusion based on such a small population, 30,000 somewhere. Yeah, so it's very early to answer that question. You ask my opinion. Yeah, my opinion, I would prefer to get vaccinated by Covaxin. The next question, view of sero survey in calculating herd immunity, as level of antibody can be raised up in any kind of infection in the order of MD. Yeah, seros. It's. See, in chikungunya, I would say yes, herd immunity would play a role. But I'm not very sure about SARS-CoV-2, because the natural infection itself doesn't give such a strong immune response. Our own lab's publication in vaccine, what we have seen is that at least 12 more than around 12 to 15% of individuals do not make any antibodies. So it's herd immunity. I doubt SARS-CoV-2 there is any big role about herd immunity. What is the next question? After COVID vaccine, many people get side effect. Will it be lifelong or short period? Yeah. Side effects. I personally had 1.5 day of headache and body pain, slight fever. My wife didn't have any. My neighbor, but since again, there has been no retrospective studies which shows that what leads to these side effects. Yes, probably people just don't are shy to take the vaccines because they hear a little exaggerated side effects from their near ones. So I think all of you should just, if you're not vaccinated, go ahead and get vaccinated. Do not worry about it, because we should all think about the larger picture for what we are getting vaccinated. Those 3 days or 2 days of side effects would not matter much. So all of you personally also should advocate for everyone to get vaccinated. Side effects would not matter. Look for the larger picture benefit. It's every research, any study is always done based on risk versus benefit. So you measure what is more. Is risks are more or benefits are more? So measure that and go ahead. Yeah, Dr. Satyendra Sinha, Sahi, you're right. When I would believe the same, it's because it's it's about host pathogenesis. That's why we have no pattern of how patients behaved patients have behaved after getting infected. There's no pattern. People have done a lot of studies on that if someone has, but yeah, there is no pattern. Dr. Satyendra Sinha is right, saying that yes, it depends on the cytokine storm, and that all again depends on what if I'm exposed to other SARS-CoV virus in my past.
Sir, there is a question from Srinivas. What is the difference between antibodies and memory B cells?
Whenever there is a what I was trying to show the slides. Fine. See, when the immune cells come across the antigen, the B cells, they produce this plasma blast. As we say, the antibodies are immediately produced. But then it goes with the healthy cells. There are certain cells which go into germinal center for future use. They are the memory B cells. So memory B cells can again produce the same Xerox copy of the antibodies which are produced earlier when there is a second encounter. So there can be millions and millions of memory B cells which would be there. But whenever there is a second encounter, you get the Xerox copies of those previously produced antibodies. It's like the machine says, go, produce the Xerox copies, but the chip is already there. So did I answer you, Mr. Srinivas?
Yes, can I speak to Dr. Kaustav Naik?
Yeah, go ahead.
Yeah, this is Dr. Srinivas. Congratulations, Dr. Kaustav Naik, because just I have seen your article which is published in Virology. Based on that,
right.
I asked some questions. Congratulations, sir.
Oh, thank you. Yeah, so that was the study we wanted to show about plasma therapy.
Yes. Just I have seen your article.
Yeah, because
very happy that you are working on COVID-19.
See, because plasma therapy, ICMR took it up and then they gave it, they didn't take it forward. One reason probably I could think why many places plasma therapy didn't work, because plasma therapy was given, they took plasma from any individual who had probably out of COVID. But what we should have done is that taken those individuals who had only, if you see my paper, we have seen that people who have antibodies at the titer 1:1000 and above are the individuals who actually have neutralizing antibodies. If the plasma of the donor doesn't have neutralizing antibodies, it's of no use for the recipient.
So can I request a small
You understand?
request?
Because
A small request. We're unable to open our article on Google. So can you?
I'll send it to Manivannan. He will forward it to you.
Yeah. Okay. Thank you very much.
Thank you very much, sir, for sharing your expertise about techniques in immunology, a snapshot with all of us.
Sir, there is a question too.
Thank you.
Yeah, I will read it out, sir. Will you suggest any ideas regarding how to approach a PhD guide after completing their Masters in Life Science and to pursue their career in research in life sciences in any national institutes?
Yeah, so see, there are multiple ways. One is most easiest way is if you have your own fellowship, like ICMR, DBT fellowship, CSIR fellowship, and there is many people do not know there is a fellowship known as Lady Tata fellowship. Lady Tata, if you Google Lady Tata Mumbai, you will be able to see. It is a private organization, but their fellowship is on a par with any other government funding agency. So you have to present, unlike other government agencies funding where you have to clear their exam, Lady Tata Junior, Senior and Postdoctoral Fellowship is based on only your research proposal. So you send them your research proposal with a letter from the institute's director that they are ready to take you as a PhD student. They will call you, an interview is held in Mumbai, they pay you for the travel to Mumbai and back. And so this is the easiest method. Then of course, now and then institutes keep calling for students for their project, project fellows. So many institutes allow the project fellows to register for PhD. So these would be the few ways to approach. What do they want? Is there a particular? What do you mean by approaching?
Sir, I think by how we should reach out by sending a mail or contacting directly in person. I think that is the question you asked first.
See, for example, your interest and the person's interest should have some amount of some amount of common link, right? Because you should be able to relate. You're going to be in that lab for the next 4 to 5 years. So you should be able to relate what you are going to do there. Or there are certain projects which have, so while approaching the prospective PhD guide, the person should probably thoroughly read that person's scientific background, because for example, when we take students here and we just gauge at the students a little bit, because what is your scientific background is there on your CV, and how you approach in doing science. Approach to life also matters a lot. How you approach to do science, how we approach to approach a problem. So it's not about just getting a very good CV and then so as a student of masters, read about the person's scientific background very well. So when you discuss, you don't stammer, or you don't get stuck with their scientific, because we don't ask direct. Many people don't ask you questions, and if you don't answer question, then they'll take you. It's not like that. So just gauge at which lab you want to join. See if you're interested in that field. So, and yes, have good scientific background of that particular field at your level. And then approach the persons. It's not that hard to get absorbed in a lab. But yes, if you have a fellowship, it's a cakewalk. If you don't have, also, it doesn't matter. If a prospective PhD boss sees a spark in the student, they might as well write a project for you. They can take and create a fellowship for you. So it doesn't really need to be fellowship leke hi jana hai. It's not like that.
Thank you, sir. Sir, people ask for your contact details to contact you further.
Yeah, I mean, Manivandan has them. He can share it.
Okay, sir. Thank you, sir. Is there any other question from the participant side? If so, you can either put it in the chat box or unmute yourself to clarify your doubts. Thank you, sir, for answering all the questions asked by your participants.
Oh, thank you.
Here the participants are requested to fill the feedback form which is put in the chat box. Thankfulness is the beginning of gratitude, and gratitude is the completion of thankfulness. Now, I request our beloved Assistant Professor of Department of Zoology, Dr. K. Kannan sir, to deliver the vote of thanks. Please, sir.
Thank you. It is my privilege to propose the vote of thanks on this occasion. First, I owe my sincere gratitude to our distinguished President, Dr. M. Marichami sir, for his blessing in each and everyday activities. I extend my heartfelt thanks to our dynamic secretary, director, Dr. C. A. Vasku Madam, for providing immense support and encouragement for all the endeavors. It's great time for me to our most respected principal, Dr. M. Lakshmanaswamy sir, for his encouragement and timely support. I'm also take this opportunity to thank our most valued session guest of honor, Dr. Joseph Noyak, research scientist, IGCP in Memory Vaccine Center, International Center for Genetic Engineering and Biotechnology, New Delhi, for sharing expertise with the enlightened our knowledge on immunological technique. Definitely, I know that all the students are getting benefited. Also, very good interaction, sir. Thank you, sir. I'm also thank Dean and Research Development and Dean of Academics for their support. I must thank for our head in charge, Dr. Binugumari Madam, who is with us all the time for encouraging us. I also extend my thanks to all my faculty members and non-teaching staff who were the pillar of the successful completion on this session on this webinar series. My gratitude to all our participants who involved made this session more valid. Thank you. Once again, I am on behalf of Department of Zoology, Ongunada Arts and Science College, on my own extend heartfelt thanks to one of all present here and encouraged to complete this session successfully. Thank you.
Thank you, sir, for expressing your gratitude through your humble words.
Before we wind up the session, we wish to say that the fifth session of the conference will be held by tomorrow at 11 a.m. and the speaker will be Dr. Ramachandran Nagarajan, Lead Scientist, Project Strategy Design, Consultant and Scientific Advisor for Bioservice Industry, Life Science, Bangalore. Title of the topic will be Pichia pastoris, a workhouse for industrial and therapeutic proteins. We request all the participants to join with us on tomorrow to enrich our knowledge.
Once again, I thank everybody on behalf of myself and organizing members, the Department of Zoology, Kongunada Arts and Science College, Coimbatore, Tamil Nadu, for making this day a great success. And let's wind up the meeting now. Thank you.